All right. So this will be our last chapter, as I've been telling you, right? This is the protein, and so we will learn about this. And just like the carbohydrates, if you look at the proteins are also we can actually regard, we can see it as a polymer, right? When you look at the polymer was you can make polymers based on the monomers. So just like carbohydrates, proteins are actually based on amino acids, right? So in order to understand the proteins, we need to understand the smaller molecules, which is the amino acids. So that's why we're going to start from the amino acids. All right. So, okay, so these are going to be our learning goals. We can skip this here. So proteins are, as you can imagine, are really important in our living cells, right? And if you look at our whole body, it's pretty much everything is based on proteins, right? All the enzymes that they are, functioning in our body is like telling what to do, right? And things like that. So everything really is like, they have very, very crucial roles, right? From transporting oxygen in the blood to aiding in the clotting of the blood, regulating cellular activities, and then you name it, right? So everything basically they involve in pretty much in almost all the things that we are doing in our bodies. So again, in order to understand the proteins, we need to understand the monomers, and in this case, the monomers are amino acids. Okay, so what are the amino acids? If you look at the amino acids are interesting in that sense. It has actually a typical structure, and this is our typical amino acids, right? This is the general structure, and I think the name is also, speaking amino acids. We have two functional groups, right? First, we have an amine group, and again, what was the amines? Whenever you have NH2, right? NH3 ammonia. This is the simplest amine, and if you replace this with any hydrogen with the R group, then you got your amines, right? So as you can see, this is your amine right here. And this is attached to a carbon, right? And this carbon has one hydrogen, and then you have an R group, okay? And so by changing this R group, right, you can actually get different amino acids, right? As far as like we are concerned, there are actually around 20 amino acids. So by changing this R group, you are actually getting a new amino acid, right? So you can put, like, let's say methyl here. This is one of the amino acids. But if you put like ethyl, that's another amino acid. Make sense? The general structure is the same. And on the other hand, if you look at this side, we have a carboxylic acid, right? What was the carboxylic acid again? Carboxylic acid is, right, whenever you have oxygen, carbon double bond, OH, attached to an R. As you can see, right, this is my carbon, this is my R group. So as you can see, it has two different functional groups on the same molecule. And this is why we call amino acids. Okay, so let's take a look at a little closer. So again, if you change the R group, then you're going to have a different amino acids. And we have, believe it or not, 20 major amino acids in the proteins. And all of the proteins that we have in our bodies are based on these 20 amino acids, right? You don't have like hundreds of amino acids. You have just 20, but by combining them all, as you can imagine, you can have, unlimited number of proteins. And that's exactly what's happening here. And depending on the R groups, right, some of them are nonpolar, some of them are ionic, and some of them are polar. So that's why you kind of like categorize them. So again, if I want to give the examples, right, if this is just a hydrogen, as you can see, this is glycine. okay? The name is glycine. If you have a CH2OH, then this is serine. And as you can imagine, whenever you have an OH, we said that the alcohol was actually polar, right? So therefore, if you have a polar group here, then this will be a polar amino acids, okay? And by the way, I'm not going to be asking you to memorize the name of this amino acids, okay? So you don't have to worry about them. And as we know, if you have a regular carbon chain, hydrocarbon chain, then this group is going to be non-polar, right? If you have just a metal here, or if you have an isopropyl group here, as you can see, these are non-polar, right? We've seen it already. So therefore, this will be a non-polar R group. And as you can imagine, you can actually have an ionic R group as well, such as like acetate, right? As you can see, this is another carboxylic acid, but if you deprotonate this carboxylic acid, as you are seeing, there's a negative charge on oxygen, right? And there's a negative charge on oxygen. So if you have such amino acids, right, because this is your R group right now, and this is your R group right now, then we are going to call this an ionic amino acids, okay? But as you can see, all of these amino acids, one thing doesn't change, right? You have an amine group here, and then you have a carboxylic acid, right? And then there's a CH in the middle. You see that, right? Everything is same, except the R group, basically. And there are actually, when you look at the amino acids, there are some essential amino acids, and these are 8 of them. And we cannot actually synthesize this in our body. Okay, so therefore, we have to actually consume these amino acids, right? We have to either eat like oil, you know, or eat like some sort of like, you know, animals and things like that, right? So that's why these are going to call essential amino acids. And these are the name of the, again, amino acids, essential amino acids, right? Again, you don't have to really know them, but as long as you know what essential amino acid means, that should be fine. And then there is another actually categorization of the amino acids, and we're going to see actually 2 different categorizations. This is one of them, and we're going to call one is a complete protein. So I think the name is, you know, talking by itself, right? Whenever you say complete proteins, it contains all the amino acids, all the essential amino acids that we need for your body. And as you can imagine, this will be coming from the plant, you know, and you know, like animal product, basically, right? If you have for instance, egg, milk, meat, or fish, right? If you consume those type of proteins, then you're going to have a complete proteins. But incomplete proteins are the one that you're going to be actually consuming from the vegetables and plants, right? Such as like cereal, nuts. Okay. And so again, if we can kind of like back to amino acids again, we already kind of like tell, right, that it has an amine group and a carboxylic acid group, okay? And so this actually allows amino acids act as either acids or base in water, okay? And this is just a technical term, right? If a molecule or if a chemical act as either acid or bases, because we never seen, right, acid and bases at the same molecule, right? We said there's an acid, there is base, but as you can see, amino acids it has the both of the functional groups. So therefore, we call this such a molecule amphoteric groups. Maybe I can show you my open. Let me just open up my report real quick. How many we have here today? 12. Not bad. You remember this chapter? I was teaching in my basic chemistry class that I'm teaching. I was actually going over just a little bit of okay. So again, what was the amino acid like? If I want to draw the amino acids, right? Right. So you have CH, right, in the middle, and then you have NH2 on the other hand, and then you have a COOH, and let's write the COH as a carboxylic acid like that, right? And then you have an R group here. And whenever you change the R group, you're going to get different amino acids, right? So again, if you look at this amino acid, right? So you have this part is actually, as you can see, this is the amine part. And this is the acid part. And amines actually, again, we didn't really learn about this, right? Because we learned what was the base again? Does anybody remember what was the base? How we name the base, or how we can tell a substance or a chemical is a base? What was the definition of base or acid? Anybody remember?
The base is giving away hydrogen, right?
Acids, right?
Yeah.
Yeah, acids are, again, the definition of acid was, it was giving H pluses in aqueous solution. And the bases were, like, if you have like sodium hydroxide, for instance, right, they were giving away OH minuses, correct? And sodium plus. And as you can see here, right, so this is actually my acid part. So this carboxylic acid here, it's not gonna mind giving up this this hydrogen. And if I can put actually another amino acid here, let's put another amino acid here, the same molecule. And I'm just gonna write it this in a different color. CH R CO OH. Okay, and again, whenever you have an amino acid, right, obviously there's going to be million of molecules, right? Million of them. It's not just going to be one. So again, this part is my acid part, right? And so when we say acid was one that can donate H pluses, correct? So here, this is my acid, okay? And again, in the Arrhenius definition of the base, we said that it can donate OH minuses. But as you can see, I think I said that there is not one definition of acid and bases, because as you can see in this case, the Arrhenius definition of base is not enough to describe why this is a base, right? Because as you can see, the NH2 doesn't giving up OH minuses. So the other definition of acid and bases, we have like Lewis acid and Lewis base, right? So what is Lewis acid and Lewis base? In that case, if the acid is donating H pluses, the base is accepting it. Make sense? So as you can see in this case, this base is accepting it because it has a lone pair here, right? If you remember the octet rule that we learned, all the elements, all the like carbon and nitrogen needs 8 electrons in their valence shell, in the last shell, whenever they make a compound. So as you can see, it has 2 bonds here, right? NH2 is like that. You have 2 hydrogens here, and it has attached to another carbon. And as you can see, we have 2, 4, 6. It needs 2 more electrons. And again, indeed, there's lone pair electrons here, 2 lone pair electrons. So again, these 2 lone pair electrons can accept, right, this hydrogen. It can grab that hydrogen. Make sense? Even though, as you can see, it's not really donating OH minuses, but we are still gonna say that amines are gonna be base. And again, this is the another definition of acid and bases. And again, this will be our Lewis base, for instance, right? Okay, you don't have to worry about the naming here, but that's what it is. So again, in reality, okay, this NH2 again can grab this hydrogen, and so I can actually put this hydrogen back here and make a bonding here, right, like that. Make sense? And whenever I do that, right, this O is going to have a negative charge, right, because we lost hydrogen. You don't have to worry about this, but we actually show how to calculate the formal charges. And now this hydrogen, right, this nitrogen, sorry, because it accepted another hydrogen, now we have how many hydrogen? We have 3 of them, right? We have 3 of them, and it's gonna accept a plus charge, okay? Make sense? And as you can imagine, if you have this molecule, you're gonna have the same thing, right? There's gonna be a negative charge here, okay? And then there's gonna be another NH3 here, and another amino acid. Make sense? Okay? So what happens if you have 2 negative and positive charges in the same molecule? Do you remember the ionic bonds, right? We said that the ionic bonds were very strong. Why? Because, like, for instance, sodium, that's right here, chlorine and sodium plus, right? Whenever they have 2 opposite charges, they can actually attract each other. Make sense? And indeed, as you can see, since we have 2 opposite charges on these molecules, they're going to be actually coming together, right? They're going to be coming together, and they're going to be making a bond between them. All right?
So, Professor,
yes,
when the base, the nitrogen, takes the donated hydrogen, does that base become neutral?
No,
It's still a base.
It becomes actually, again, you don't have to worry about this, but it becomes, we didn't learn this. We call this actually conjugate acid, but you don't have to worry about it.
Okay, yeah,
okay?
yeah, okay.
Okay, you don't have to worry about it.
Yeah.
And then we are gonna actually call, this molecule then. There's a term for this, okay? We're gonna call it as, this is how I remember actually, Twitter, right? So it's not gonna be Twitter, but it's gonna be Zwitter. It's a funny word, but it is what it is. Okay, so Zwitter, it's gonna be Zvitarian. Okay, so whenever you say the Zvitarian of amino acid, it means that it is the protonated and unprotonated version of the amino acid. Protonation means losing the hydrogen or getting the hydrogen. Make sense? Protonation is, proton is, again, we call this a proton, right? At the end of the day, this is a proton. H plus is a proton because it has only 1 proton and no electron. And that's why we call H plus as a proton. Okay, let me go back to my slide again. And again, we are gonna call this, right? If you have such a molecule, right? If I ask you to Zvitarian of an amino acid, right? Then what you have to do is you have to take away the hydrogens here, right? There was a hydrogen there. You have to add these hydrogens to other side here, basically. Make sense? That's what it is, right? So there's no hydrogen here, and you're adding that hydrogen to amines here, okay? And then we're gonna call this actually as Zvitarian, all right? And I'm not sure if anyone here using any protein. Does anyone use protein here?
Yeah, just your normal, go to gym supplement protein.
Yeah. I'm talking about those things. Like, and does anyone doing what do you call that?
Wheat powder.
Yeah. So whenever you are consuming those proteins or amino acids, what are they? Are they liquid or are they solid?
Well, they're solid powders that we mix with water.
Exactly, right? They are solid powder. And have you ever think about it why they are powder, Carlos?
Yeah, because they're taking proteins and then dehydrating the proteins.
Okay.
Whatever.
Yeah.
Like eggshell, they will actually dehydrate. If you have an egg-based protein powder, they take the egg and they process it, make it a powder, they dehydrate it, and then you mix it and they add some other food flavoring in there so it doesn't taste like crap.
I know, but at the end of the day, whatever you are eating, all right? Again, I never, eat those, but at the end of the day, whatever you are eating is some sort of amino acid, right? It's like one of these molecules, right? I don't know what the R is going to be, but there's going to be different R, obviously. Obviously, you are eating like one of those 8 essential amino acids. But my question here is, if you look at the corresponding like compound, like let's say you have 1, 2, and let's say there's a metal here. Okay, I'm just making this up. Let's say hydrogen. Okay. Okay, that's a hydrogen. The R group is hydrogen. And if you look at the molecular weight, right? Let's say we have 1, 2, right, and 2 carbon. And then you have N, hydrogen, and there is another carbon here, let's say. I'm just making this up, okay? Like, I'm just like, compare this molecule with another molecule. And then you have 2 O group, right? Let's say OH here and OH here, right? Again, just the same molecular weight, but it has and then let's put another nitrogen here somewhere. Again, I'm not sure if there is such a molecule, but I'm sure you can make this molecule. It's not going to be a problem in H2, let's say, okay? Right, so as you can see, the molecular weight is going to be more or less the same. So my point here is, Carlos, right, if you look at this molecule, I can guarantee you, right, because it has only 3 carbon, right? It has only 3 carbon.
Right.
When you look at the hydrocarbon, 3 carbon, what is it? It's a propane, right?
Yeah.
And propane is, what is propane? Is it liquid or is it gas?
Gas.
It's a gas, right? And let's say you are putting like OHs here. And so I can guarantee you that this molecule is going to be, most likely, it's going to be either a gas or liquid, right?
Right.
So when you look at the amino acid, on the other hand, they are solid, right?
Right. Yeah.
So why they are solid?
They had to add more carbon. So you got 1 to 4 carbons are normally gases. So if you add another carbon, it becomes a liquid.
Yeah, but I mean, again, as you can see here, we are not really adding carbon here, right?
Right.
It's the same carbon. So what makes molecules liquid and solids are, how they are tied together, right?
They gotta be emulsified with something else.
Right? So like what makes the molecules are liquid, solid, and gas, right? If it's a gas, it means that the molecule there is no interaction between the molecules, right? That's why the propane is a gas, because the molecules are, right? This is my propane, right? 3 carbon and 3 carbon, right? There is no interaction between them, right? As you can see, there's no interaction, very, very limited interaction. But whenever we add OH here, right, let's say OH, and make a propanol, as we know, propanol is, right, isopropanol is liquid. Why? Because now OH is polar and you can have an interaction between them. Again, when there's an interaction between them, then they're going to come together, right? They're going to come together. They're going to sit together, right? So as you can see, if you look at the amino acids, right, even in the neutral form, right, we are going to have this Zvitarian. And so therefore, as you can see, the NH3 is going to be protonated, OH, this acid is going to be deprotonated, and there's going to be a strong interaction between this plus and negative charge. They're going to be coming together, okay? And again, as you can imagine, there's a million of molecule, and there's going to be very, very strong interaction between these molecules. And so that's why it's going to become solid now, right? Because you have to break up this bond in order to get liquid, right? You need to separate those molecules. Then it's going to get liquid. If you separate more, it's going to become gas. But as you can imagine, now they are like this, right? Like if you look at the sodium chloride, is sodium chloride is liquid? No, right? It's a solid.
It's a
right?
salt,
Why?
yeah.
Because it's an ionic bond, right? Ionic bond is you have a negative charge and plus charge, and it's very strong. Again, this is not an ionic bond per se, but it's like almost like an ionic bond between these two molecules, right? Because we have a one negative charge on the one side and one positive side on one side, right? It's going to be like this, basically. Like we have molecules like that tied together, right? One side is plus, one side is negative. One side is plus, one side is negative. You're getting right? Okay. So that's why then the amino acid is going to be again solid at room temperature. And this is the reason why the chemists actually come up with this Zvitarian, right? That's the only explanation why the amino acid is going to be actually a solid. All right. So again, in the in the neutral molecules, right? Even it's the neutral molecules, it's going to exist as a Zvitarian, okay? So that it can have a dipolar, action between this negative charge and positive charge, right? If you bring in other amino acids here, again, this is going to be a plus charge here, negative charge here. There's a strong interaction, and you can bring in another amino acid here, as you can see. Now there's going to be negative charge, right, and the plus charge, etc. Okay, so now we can come to, again, just like we've seen in the whenever we learn about the carbohydrates, right? We said that if you bring the amino acids together, then you're going to get your proteins, right? So as you can imagine, whenever you have two amino acids, and again, this is one of the amino acids. I hope you can see it. Maybe I can blow this up. Right, so this is my, what color is this? Oh, this is white. So as you can see, this is my first amino acid, and this is my second amino acid. Again, the name is glycine, and you don't have to worry about it. So if you look at this, right, what functional groups you are seeing here? Does anybody remember this? Carbon attached to a nitrogen. What do we call this functional group? Anyone? Manuel? Team? Amy? Jesslyn?
Is it an amine or amide?
Yes, not amine, but an amide, right? Amide. Okay.
I remember there was two with nitrogen.
Yeah, I mean, again, it doesn't matter if it's two hydrogens or one hydrogen or no hydrogen, doesn't matter. But as long as you have, again, nitrogen attached to a carbonyl, then we're going to call this an amide. Okay. So, and if you can remember, you can make the amides by mixing up the carboxylic acids, right, with an amine, right? If you can remember that. And again, we are not going to go into mechanism, but in this, reaction, we are going to be actually, as you can imagine, we can go back here. We are going to be actually losing this water molecule, okay? And we are going to make actually, this is going to be called a peptide linkage. And at the same time, the peptide linkage, right, is nothing but an amide linkage too, right? So as you can see, you are putting like two different amino acids, and then you are making an amide basically, okay? And during this process, we are losing a water, obviously, okay? So again, this is going to called a peptide bond. And as you can imagine, if you have a proteins, you're going to have, you know, thousands of this peptide linkage between the amino acids. Okay. And as we've seen again in the carbohydrates, right, you can actually have dipeptides, right? When we say disaccharides, what was the disaccharides? You have two carbohydrates, right? So if you have a dipeptide, it means that you have two amino acids forming a peptide bond. If it's a tripeptide, it's a three. Polypeptide, you have many. Okay. And again, you don't have to worry about these two, but as you can imagine, right, you can actually, whenever you have two amino acids coming together, you're gonna have kind of like an order, right? Like if you, where is it? Yeah. So if you have this amino acid, which is glycine and alanine, right? You're gonna have two possibilities. You can put the glycine first and alanine second, right? Or you can put the alanine first and glycine second. Makes sense? Because at the end of the day, you are making this bond between them, right? So since you have in both ends, you have amines and carboxylic acid, right? That's why you can actually, you know, as if you are making a puzzle, right? You can have two options of putting the first one first and the second one second, and vice versa. Okay. Okay. And again, this is the one of the last definition of proteins, like how we are classifying. And the first one is a fibrous proteins, and these proteins are insoluble in water. Okay. And why they are insoluble in water? Because they have to be insoluble, right? Because if you think about it, right, like keratin, for instance, right, which are found in our hair and nail, they are insoluble, right? If these proteins will be soluble, then this will be melted away, right? We are not going to have any nails, and we are not going to have any hairs, okay? And then obviously in our body, there are going to be some other proteins, because our body is consists of 95% of water, and some of these proteins has to be soluble in water, otherwise they cannot function. And if you look at like pretty much all the enzymes, right, like hemoglobin, right, and lipoprotein, and these all proteins are going to be actually in our blood, and they are soluble in water. Okay, again, fibrous proteins are insoluble. Global proteins are soluble. Okay, these are two types of proteins in our body. And again, the function, I don't have to talk about the functions, you know, as you can see in our every single one of the systems, right? From blood to, again, to digestive, from respiratory to anywhere, right? So there's proteins everywhere, basically. Okay, so now here's the critical part. So since we are actually now bringing this amino acids, right, we are going to actually learn the structure of the proteins. Okay, and this is actually where things get a little excited, and we are going to actually learn that there's going to be 4 different types of structure of the proteins, right? Because remember, whenever we were talking about the isomer, right, if I ask you the proteins, for instance, right, let's say hemoglobin, and in hemoglobin there are 60 hydrogens, I'm just making this up, and you know, 100 hydrogens and 10 nitrogens and 5 oxygens, right? And again, we learn in organic chemistry that, right, remember the first chapter, this means nothing in organic chemistry, right? Because again, what is the structure of this, right? How you are like, how did you put together all these carbons, nitrogen, and oxygens, right? So that's why you need to understand the structure of the proteins. And the first one is, it's a primary structure. And the primary structure is nothing but a basic sequence, right? An order of the amino acids which makes up the protein. If I want to give you actually an example of hemoglobin, right? So if you look at the hemoglobin, there are 146 amino acids, right? So different amino acids, 146 amino acids. Some of them might be same, some of them are different, but in total, there's 146 amino acids. So the primary structure is telling me the order, right? So I'm like attaching A to B to C to A again to C to D, etc. Right? So there are 146 of them, and in the primary structures, I know exactly which one is attached to which one. Make sense? So this is the again the structure of the hemoglobin. So in the primary structure, you know the order, right? So the first one is valine, the second one is histidine, the third one is this, third one is trioptan, etc. As you can see, you know the order, right? So if I change, for instance, let's say you want to change this one to something else, right? Some other amino acids, then this is not going to be hemoglobin. Okay, this is going to be something else, right? Obviously, if you just change one of the amino acids in this chain, you kind of like mess up the protein, right? You're getting a defective protein, or it's not going to be hemoglobin anymore. It's going to be something else, and it will not be able to function as hemoglobin. Okay, this is how precise it is, and we're going to call this a primary structure of hemoglobin. So the secondary structure tells of how the polypeptide chain is arranged in space. Obviously, this is like on paper, right? And again, this is actually a molecule, and I'm just putting the name here, but obviously this molecule, as we've seen in 3D, right, it's gonna have like, this is gonna be in 3D space, right? So the secondary structure is telling us then how this polypeptide chain is arranged in the space, okay? Right? And if you look at it, then you're going to have this different type of secondary structures, such as alpha helix, beta pleated sheet, or triple helix. And these are going to be the examples, if I can show you. Again, these are not so great, but you can actually get the idea. I have actually, let me show you in my other PowerPoints that I was teaching. I didn't use the slides, but yeah, so this is the, again, kind of like the secondary structure, right? So this is a primary structure, as you can see. This is the order, right? But in reality, this is going to be actually in 3D, right? So therefore, you're going to have some sort of like a shape, because at the end of the day, these are going to be interacting with each other like that, right? There's going to be interaction, and this interaction is going to kind of like dictate how this molecule is going to shape up. It's going to be like a coil, or maybe like a spiral helical arrangement, and etc. Right? So this is where the secondary structure is going to come in. Make sense? And as you can see, you can have actually, you know, a different type, right? You can have, again, if there's an interaction between these two, and these two, and these two, and there's going to be a helix structure, or it can be a kind of like a sheet too, right? Like if there's an interaction between these whole chain, right? This is again this is my chain, and if there's an interaction between them, then it's gonna look like a sheet of paper, or it can look like a helix again. Okay, makes sense, right? Everyone? All right. All right. So we are learning again the structure. So this is secondary structure again. These are another examples. And this is again triple helix of the collagen and other proteins. And so what about the tertiary structure? So the tertiary structure is again, this three-dimensional structure of protein is called tertiary structure, right? Again, at the end of the day, we have this helix, right? But as you can imagine, this helix is going to be in 3D, is going to be folded in a very specific way. I think I have a better picture on the other slide. You are seeing it, right? As you can see now, this is my alpha helix. This is the secondary structure, but because again, this is large molecule. It's a very, very large molecule, okay? And so it's kind of folded, right? It's kind of folded in a very specific way, okay? And whenever it folds, then we're gonna have a tertiary structure. And again, this is the tertiary structure of the one of the protein here, myoglobin. And as you can imagine, it has to fold it like in this way so that it can function. As you can see, there are some cylinder here, and again, this is like some sort of like a molecule that's a chemical, right? And this protein, again, whatever it does, I don't know exactly what it does, but as you can see, it's binding this molecule, right? It has some sort of like a cavity here, as you can imagine. So because of this cavity, it can actually target this molecule. Make sense? Because if the protein, if this protein doesn't fold it in a this tertiary structure, then it will not be able to interact with this molecule, right? It's not going to have this cavity, and then it will not have the functionalities, even though, again, it will have the same structure. Make sense? So it has to, again, fold it in a very specific way. So it has to have a very specific tertiary structure so that it can actually, again, does it function. And how we are actually learning this, again, this is a very long process, as you can imagine, right? You are starting from the primary structure. You are first determining the primary structure, and then you are determining the secondary structure. And then you have to actually get crystals out of these proteins. And once you have your crystals, the X-ray crystallography guys are studying it, and then they are getting the 3D structure of this protein. Okay. And once you do that, then you can actually publish your results. But again, this is a long process. And we don't know, again, even though we know a lot of proteins that we know the sequences, but there are a lot of proteins still, and people are studying every day, obviously, to get the tertiary structure of the protein, right? Get the X-ray structures, and so that they know exactly how this protein is folded.
So let me ask you, with the with kidney stones, bladder stones, isn't that the body wasting protein that's not being used in the body? That's why people get kidney stones and bladder stones.
Yeah, I'm not sure about that.
Because they're crystal-like, they're like salts, but they're not. It's waste. Yeah, I don't know. I was just asking a general question.
Yeah, again, I'm a chemist, so
yeah,
that's I really don't know the reason, but I think, you know, it can be some other reason too, because those stones are stones, so those are like most like salts, right?
right.
So most likely your kidney is not filtering, properly, as you said, and so the sodium chloride or other salts and things like that, they are not getting into your urine, I guess, right? So that it stays in the kidney, and then as they stay, it accumulates and accumulates and accumulates, and then it becomes a stone.
Yeah. Okay.
Yeah, but probably they are not proteins, most
Right.
likely. They are probably like insoluble, like salts and things like that.
Professor, so
Yes.
these proteins, they're like this is all essentially like the same. It's the same protein, just different ways of looking at it, like the primary, secondary, and tertiary. It's not.
It's this. Yeah, it's the same protein, right? Like here in the
Yeah.
first one, this is the primary. This is still, let's say, again, one of the molecule. What in the other structure we said, let's say this is the hemoglobin, right? This is the
Right.
primary structure of hemoglobin. This is the secondary structure of the hemoglobin. Now, because this is in 3D, okay? This is the
Right.
secondary structure. This is still the same protein. And if you come to tertiary structure, this is still hemoglobin, but this is the tertiary structure of the hemoglobin.
Right. Okay. And so if you were to look at it like in a microscope or something, or in your body, it will exist as looking like the tertiary, correct?
Correct. But in the microscope, again, you will not be able to see this structure. Okay.
Okay.
Because
Even
in the
with like an
microscope,
electron microscope or something powerful?
even, yeah, like again, they are looking the structure on the X-rays, right? So this is again, whenever the molecule, because whenever you dissolve this molecule, and again, I'm going to show you that why it happens, right? Whenever you dissolve this, right? Because there are a lot of interaction between these. Obviously, there are like thousands of carbons, nitrogens, oxygens, and there are thousands of linkages. Again, like here, for instance, right, we have like a disulfide linkages. If there's a sulfur here and sulfur here, it kind of like, again, you don't have to know this, but it kind of like binds this together like that, okay? And this dictates the shape of the molecule, right? and the structure of the molecule, tertiary structure of the molecule. And again, the only way to get this is get the X-ray crystal structures.
Okay.
So you need to make a solid, right? You need to make a crystals out of this protein, and then you have to send it to a crystallographer. That's the only way to get the exact structure of the any molecule, basically, right? Whether you're talking about like just a water molecule or any molecule, you need to have an X-ray crystallography to take a look at it. Okay. And again, so this is tertiary structure, and I'm going to show you one more. And again, whenever you have these tertiary structures, and again, this is the myoglobin molecule, right, in tertiary structure. And again, these are all the interactions that we are talking about, right? Hydrophobic interactions, hydrophilic, salt bridges, hydrogen bond, disulfide linkages, right? These are all happening in the same molecule, so that I'm making a kind of like a bond, intramolecular bond in the molecule, so that the molecule is like folding in a very, very specific way, okay? And some proteins, right, this is the last structure, some proteins, and these proteins are very large proteins, they're gonna have quaternary structures. And what's quaternary structure is, this means that they're gonna exist as a group of 2 or more polypeptide chains. In other words, you are kind of like bringing those tertiary structures together, and then you are making a one large quaternary structures. And if I can give you an example, right? So this is the actually quaternary structures of the hemoglobin. Now, as you can see, this is one of the subunits here, right? This it's hard to see, but it's not so bad, right? This is like, again, one of the tertiary structures, as you can see, and there's another tertiary structures here, right? So they are coming together, right? And then they are making a big quaternary structure of the proteins, okay? Again, as you can see, the molecule is comprised of 4 subunits, 2 alpha chains, white block, and 2 beta chains, gray block. And the heme group is shown as a light red. It's the iron containing structure. Okay, so as you can see, right, this is the hemoglobin. And as you can see, the hemoglobin is a very large molecule. So in the middle here, right, if you have an iron, again, we are not showing the iron here, there's an iron here. And so iron molecule kind of like binds with this protein, so that it can actually carry out oxygen molecule, right? So the oxygen can bind the iron from top, 1 oxygen from top, and 1 oxygen from the bottom, like that, so that it can carry out the oxygen in our body, okay? So this is the last structure of the protein. Makes sense, right? So the quaternary structure is kind of like putting together 1 or 2 or 3 tertiary structures all together. All right. Any questions, everyone? All right. So we have also called, if you can remember, I'm going to take back to our very, very first chapter. If you can remember, we talk about eggs, right? I think the other slides is better. Let's go to other slides. If you talk about the eggs, let's say eggs, and then you cook the egg, right? Energy is, again, you're cooking it. And then you are getting a what you call it, cooked egg, right? And so this is cooked. And remember what we did? What did we say? Does anybody remember? Does this process is a chemical process or physical process? Physical change or chemical change?
Chemical.
Anybody remember? Huh?
Chemical.
Okay, chemical. Why is chemical?
Because you're breaking down the proteins with heat.
Well, as you can see, we are not really breaking down the proteins, right? This is protein, and as you can see, whenever we have a protein, right, this is like a long molecule, right? It's the same molecule, as you can see. This is the same molecule. We are not really breaking down. I have, as you can see, 2 of them, right? If I have a carbon, so the primary structure is the same, as you can see, right? Primary structure is the same. right? Primary structure is like you attach to one amino acid to second, third, fourth, right? So the essentially it's the same, right? If you have 10 carbon here, I'm just making this up, you have 10 carbon there, and like nothing changes. It's the same molecule, but why it's chemical? Right? Because this is chemical because again, we are kind of like breaking down the three-dimensional structure of the protein, right? So we are actually, this is folded protein. And whenever you break up those interactions, again, why? Because we have interaction between these two, right? These two, like there are thousands of interactions, but because once we apply energy, once we apply heat, we are breaking up those interactions, and it's actually getting defolded, right? And so we call this a denaturation, okay? And as you can imagine, once the protein is denaturated, it lost, right? It loses its function, because as you can, as we said, right? In order for this protein to function, it has to be folded very specific way. If it's denaturated, if it's defolded, then this will not be actually function anymore. And in some cases, this process is actually reversible. You can come back to here again. But in some proteins, right, if you apply heat, for instance, as we can see in the egg example, you cannot go back to your first state, right? Once you apply heat, it's cooked egg. You cannot go back to it. And that's why it's chemical change. Even though the way that we understand, right, the number of the carbon, hydrogen, oxygen is the same, but because it lost its function, then it's going to be a chemical change. And we call this actually a denaturation. Okay, and you can actually do this like in different ways. For instance, this is energy. First one is energy. And the second one is you can actually dissolve it in water, right, or some substance, or maybe in alcohol, right? Remember, like whenever we use sensitizer, right? We use what? We use wipes, right? And in this case, like in the virus case is different, but right, if you have a bacteria, for instance, let's say you have a bacteria, and whenever you have a bacteria, right, the proteins inside the bacteria is going to be the one that's going to be harming you. And so whenever you dissolve, right, whenever you wipe with isopropanol or whatever, right, the wipe has, so you're breaking up those bonds between the proteins, and then you are defolding the proteins of that bacteria, so that it's not functioning anymore. Make sense? That's why whenever we want to sterilize things, right, we want to heat it up, or we want to like dissolve in alcohol and things like that. That's one of the reasons too. All right, again, this is called a denaturation. And again, if you denaturate, then it becomes an inactive protein. It's not active anymore. Okay. So basically, the primary structure is the same, right? But you are actually destroying the tertiary structure. Okay. All right. So if a protein unfolded can be refolded, again, this can be a reversal, but in some cases it cannot be. Okay. And again, heat and action of solvents denaturate proteins. And these are the things that we mentioned. And you don't have to worry about the proteins test, even though I'm going to give you one lab. It has a kind of like a test for the proteins, but for the lecture, you don't have to worry about it. And I'm not going to be asking this test, okay? And again, these are biuret tests. Biuret tests take the presence of the peptide bonds, but we don't really, I don't really cover this test. Okay, now I have these questions, kind of like wrap it up, okay? So when you look at the first one, do you have any questions, by the way? So if I ask you, if you look at the first question, let's actually expand this. So which one is do you think the amino acids here? In the first question. I'm trying to find my amino acids. Which one is my amino acids?
B.
B. Everybody agrees? Why it's B? Tim?
Because I see the carbolic acid on the right side.
Yep, yep.
Okay. And again, it has a carboxylic acid, but that's not the only reason, right? What else?
And you have the amin on the other side.
Yeah. So you have the amine here, right? And they are attached to same carbon here, as you can see, right?
Mhm.
So therefore, this is my amino acids. Again, I don't know which amino acid it is, but it's an amino acid, right? As you can see here, this is a amide here, right? This is not an amino acid, and there's no carboxylic acid. And same thing, there's no carboxylic acid. This is amide, and this is just an amine, right?
Mhm.
This is just an amine. Okay? Makes sense, right, everyone?
There's mud.
All right. So what about this one? So if you look at these, right, we said that you can actually, this is my tripeptide. Well, I kind of like give it away, but so if I ask you, this is my one of the proteins, right? Let's say one of the proteins. And how many amino acids that you see here that you put together? Can you tell me? How many amino acids that we need to make this peptide linkage?
Yeah, this is one.
They're going to be 3, right?
3?
Because you have 3 R groups.
Okay. So you're looking at the R groups, or so whenever you have
Yeah.
such, huh? Say it again.
Yeah, no, I mean, that's just how I looked at it because, you know, you have the 3 R groups coming off the top. So,
Yeah. Well, I mean, that's one way, but I think that might be a little bit misleading. So the best way to determine is look at your amide linkages, right? Amide linkages, because that's how you make the proteins, right? So amide linkages is again nothing but a carbonyl attached to a nitrogen, right?
Mm-hmm.
So as you can see here, you have one here, right?
Yeah.
And there's another one here. So it means that, right, you have to break up these amino acids from here, right? Like that. This is 1, and this is 2. You see that, right? Okay, so therefore, this must be your first amino acid, this must be your second amino acid, and this must be your third amino acid. Make sense? Right?
So the last one has a carbonic acid on the end,
Yeah.
but there's only one of those. So
Yeah, it doesn't matter. Again, you're putting them together, right? You're putting them together.
uh-huh.
And yeah, so this is actually coming from, again, right? You have this one, OH, CH2, C, and whatever that group is, CH2, and then you have a phenyl group here, right? And so initially you have this, right, Carlos, NH2 like that, okay?
Right.
And this is one of the amino acids that you use. And then you are linking this with the second one, make sense? And then you're linking that with the first one, okay? And so that you can get this amino acid, basically. So you have 3 amino acids. So you can tell me, right, if I give you the primary structures, then you can tell me how many amino acids is there by finding the amide linkages, okay?
Okay.
Make sense?
Yeah.
All right. So what about this one? If you look at this structure, is this a primary structure, secondary, tertiary, or quaternary structure of the protein?
That's the secondary.
Secondary, right? Again, this is another alpha helix, and this is like how the primary structure is actually seen in 3D, right?
Correct.
So
Yeah.
it's again, because of these interactions, like there's the oxygen here, probably this is an oxygen, and there's the hydrogen there, and there's a kind of like a bonding between them, right? Like a little interaction there and there and there and there, so that it kind of like, again, folds like that in a kind of like an alpha helix, okay?
So you don't
That's all.
consider them turns as folds.
Well, I mean, this is, again, this is fold, but if you have larger, right, this is just the one part.
Okay,
Right?
all right.
But if you can imagine, there's going to be larger and larger molecule, and then you're going to have alpha helix here,
Yeah.
and then you're going to have another alpha helix there, and as we've seen, it's going to fold it in a very specific way like that, right?
Okay. Yeah. Yeah.
So this will become a tertiary structure.
Tertiary structure.
Yeah.
Yep.
Okay.
Okay.
All right. And that's the end, everyone. All right. So any questions, any concerns? So that will be the end of this chapter, and actually end of the course, to be honest with you. And so I'm going to actually publish the quizzes today for this chapter. And then we're going to have one more quiz, again, to cover all the organic chemistry, the last part of the whole course. And that's going to just wait as a quiz again. So, I mean, I can publish them both if you want to, but we will be finishing 1 week early, right? Official finish is May 8th, I believe, right?
Yes.
What is the last day of school? May 8th, right?
May 8th.
Yeah. So it's up to you guys. Do you want to meet again next time, or are you guys fine?
Let me ask you, you said you're going to post a lab as well?
Yes, I'm going to be posting a lab. There's one lab that kind of like covers the carbohydrates, proteins, and lipids. It's a very simple lab. Again, it's like showing you how to detect which one is which, like lipid test, like carbohydrate test and things like that.
So is the, now that you have the short quizzes for each chapter, they're still timed, is the review going to be timed as well?